show you the setup for the nerve cell communication activity. So here we've got two neurons. We have a sending neuron and a receiving neuron. So one of these is sending the signal to the next. So this is the sending, this one is the receiving. So the first part on page 1, you're supposed to label the different boxes that are on the neuron. And so I'm going to go through those. So we have what's called terminal branch, or axon terminal is what we've been referring to it as. That's going to be this extension coming off of the axon. The myelin sheath is this wrapping that goes around the neuron. And so up here, that was going to be referring to this brown surrounding around that. But to save some space, I've kind of condensed our neurons here. So I'm just going to put it right here, kind of underneath where the axon is going to be. So the axon is an extension that comes off the cell body, and it sends signals away from the cell body. The dendrites are the extensions or processes that come off the cell body and receive signals, so it goes there. Receptor proteins are the proteins on the end of the dendrites that are going to receive the chemical signals. They have specific shapes. The cell body, of course, is this main bulk of the neuron, so we're going to put it there. The vesicles are the blue dots that you see in the terminal, so they are kind of round bubble-like structures that hold neurotransmitter. And then the nucleus is going to be that purple dot that you see in the center of the cell body. And then the synapse is the space that's in between those two cells. On page 2, you need to identify the different functions of each of these structures. So I'm just going to go through them. Stores and releases chemical signal molecules. That's going to be our vesicles. Conducts electrical signals called impulses. That's going to be our axon. It's going to send that signal down to the next cell. Controls life's activities. That's the nucleus. The nucleus contains the DNA, which is going to determine all the different processes that the cell does. The gap between two neurons would be our synapse. Because again, notice here membranes do not touch, but they come close together. Carries out life activities would be the cell body. Sends chemical signals to another neuron. That's going to be our terminal branch. So once that signal reaches the end of the axon, that terminal branch then will release the neurotransmitters into that space. Attaches to chemical molecules. That's going to be your receptor protein. Insulates the neuron. It speeds up the conduction, would be the myelin sheath. And then receives chemical signals. That's going to be the dendrite. Okay, and then the next step, you are to figure out between the pink, the yellow, and the white neurotransmitters, which one of these would be the appropriate neurotransmitter to continue the signal on these neurons. So I'm going to place these neurotransmitters next to the receptor protein so you can determine which one would fit that shape of the receptor protein. Because remember, these are like keys going into a lock. They need to fit perfectly in order to send the signal on. Okay, hopefully you thought the pink. Even though the yellow one also has some lobes, the pink fits the best into that shape. Okay, so we're going to place pink neurotransmitters inside our vesicles here. Okay, so when a neurotransmitter comes and it binds to these receptor proteins on the sending neuron, an impulse is generated. And so as you travel down the length of that axon, again, notice the inside of the axon is negatively charged, outside is positive at rest. But when depolarization occurs due to an impulse, the charges flip. So where that depolarization wave is going, it's now positive on the inside, negative on the outside. So we're pulling that impulse down along the neuron, and when it reaches the end of the terminal, those neurotransmitters are released into this synapse or space, and they're going to bind with the receptor proteins on the next neuron. Okay, and once that happens, then this neuron is stimulated, and so sodium ions start moving in, and as you have that huge influx of positive ions, again, the impulse travels down the neuron along that axon to the next axon terminal where those neurotransmitters would get released. The last part I want to show you is just, sometimes we will either take different pharmaceuticals or other drugs can mimic the shape of neurotransmitters. So I have a green bead that looks very similar. Its shape, or it is the similar shape as the same shape as our pink neurotransmitter here. So if you were to take a pharmaceutical or other drug and it has the same shape, it is possible now that this neurotransmitter would also be able to send a signal. Okay? And it may be that the brain is not telling our body to do that, but because this pharmaceutical is present, it's going to allow that process to continue. So keep that in mind when you are answering the